Hey there, science fans! Today, we're talking about something amazing. They are called enzymes. Wow, enzymes do a lot of work inside living things like you and me. Think of them as tiny, super efficient helpers. But what exactly are they made of, you ask? They are special proteins. Yes, proteins. These proteins are incredibly important for life. They are like the construction workers and the managers inside our cells. They make sure everything runs smoothly. Science! Now these proteins are built from even smaller pieces. These tiny building blocks are called amino acids. Imagine a very long necklace made of different colored beads. Each bead on that necklace is an amino acid. There are about 20 different kinds of these amino acid beads. They link together in a very specific order, one after another. This order is like a secret code, a special instruction. It tells the protein exactly what its job is and how to do it. Pretty cool, huh? These long chains of amino acids don't just stay as straight lines. Oh no, that would be too simple for these amazing molecules. They fold up, they twist and turn into complex three-dimensional shapes. Some parts of the chain might twist into a spiral. We call these spiral shapes helices, like a tiny, tiny spiral staircase inside your cells. Other parts might flatten out into broader sections. These flat parts are called sheets, like a carefully folded piece of paper. These specific shapes are absolutely crucial for how enzymes work, and there's even more to their fantastic structures. Enzymes also have parts called loops. These loops are flexible segments that connect the helices and sheets together. Some of these loops are found on the outside surface of the enzyme. We call those exterior loops. Others are tucked away on the inside, forming interior loops. These loops are not just random bits, they have important jobs. They often help the enzyme grab onto other molecules, the ones they work with. They are like the enzyme's very own hands and fingers. So we know enzymes are proteins with cool shapes, but what do? Enzymes have a super important job in chemistry. They are catalysts. Now, what in the world is a catalyst? A catalyst is something that speeds up chemical reactions. Things happen much faster when an enzyme is around. But here's a key thing. Enzymes don't directly change the reaction kinetics in terms of equilibrium. They don't alter if a reaction can happen, or what the final balance of products and reactants would be. They just provide a quicker path, like a chemistry shortcut. Lowering activation energy. They make reactions go zoom instead of slow. Now, how do scientists come up with names for all these different enzymes? It's not random, I'll tell you that. Scientists love order. Enzyme names usually give us a big clue. They often tell us about the specific molecule the enzyme works on. We call this molecule the enzyme's substrate. So the enzyme's name points to its partner in the chemical dance. It's like naming a chef the pasta maker if they make amazing pasta. It's a very logical system. Smart. Let's look at some real-life enzyme names. Consider this. An enzyme called phosphatase. What do you think it does? It works with phosphate groups. It helps transfer them or remove them. A carboxylase deals with adding or removing carboxyl groups from its substrate. An oxoreductase helps with oxidation and reduction reactions. These are reactions where tiny particles called electrons get moved around between molecules. A demethylase will remove a methyl group from a molecule, and an alkylase might add an alkyl group to its specific target. See the pattern emerging? And the list goes on. There are so many fascinating enzymes. Isomerase enzymes are like molecular magicians. They take a molecule and rearrange its atoms to form an isomer. An isomer has the same atoms, just connected differently, creating a new shape or structure. Think of it like having the same Lego bricks but building a different model entirely. Enomerase isn't a standard enzyme class name, but perhaps you're thinking of enzymes that create or act on enols or enoates, which are specific chemical structures. And a big clue for many enzyme names is the ending ase. If you see ase at the end of a biological molecule's name, it's very likely an enzyme. The name itself is like a little instruction manual for its function. So why all this fuss about knowing the enzyme's name? Well, understanding the name is your very first important step. It's like knowing a person's job title before you ask them for help. If you know someone is a plumber, you have a good idea of what they can fix. Similarly, understanding an enzyme's name gives you a fantastic head start. It points you in the right direction in the world of biochemistry. It tells you what kind of chemical action to expect from that enzyme. This is incredibly helpful for scientists studying life's processes and reactions. Once you've got a clue from the enzyme's name, what comes next? You look closely at the chemical reaction it's involved in. Every chemical reaction has starting materials, the ingredients for change. We call these the reactants. They are what the enzyme will chemically modify, and every reaction has ending materials, the things that are made as a result. We call these the products. These are the results of the enzyme's dedicated work. You need to carefully identify what the reactants are at the beginning, and then you need to see what the products become after the enzyme does its job. Then, you become a detective. You analyze the changes. What exactly happened to the reactants to transform them into products? Did the enzyme help add a new chemical group to the molecule? Or did it help remove a chemical group from the substrate? Perhaps it helped break a specific bond, or form an entirely new one. Or maybe it just rearranged the atoms within the molecule like an isomerase would. For example, if you see a phosphate group being added to a sugar molecule, and the enzyme involved is a type of kinase, it all starts to make perfect sense. You're connecting the name to the specific chemical action. Science in action. You also want to look at the type of change occurring in the reaction. Is the chemical reaction a reversible one, for instance? That means the reaction can go forwards from reactants to products, but it can also go backwards from products back to the original reactants. Or is the reaction essentially irreversible? This means it mostly just goes one way, towards products. Some enzymes are great at helping reactions go in both directions depending on cellular needs. Others are designed to really push the reaction forward, making it essentially unidirectional. Knowing this tells you even more about the enzyme's specific role and regulation in the cell. It's all part of figuring out the big, complex, and beautiful puzzle of life. All right, everyone, let's wrap this up and bring it all together now. Enzymes are truly protein powerhouses, doing amazing and vital work in our cells. They are carefully constructed from long chains of amino acids, their building blocks. These chains fold up into very specific and complex three-dimensional shapes. These shapes include those cool spiral helices, flat extended sheets, and all-important flexible loops. These structures aren't just random, oh no, they are perfectly designed for function. They are designed to do their specific chemical jobs with incredible precision and efficiency. Their unique structure is what allows them to perform incredible chemistry. Science is so cool. We've learned that enzymes are biological catalysts. That's a big deal for living systems. They make chemical reactions happen much faster than they would on their own. And they do this without being consumed or permanently changed in the process themselves. Their names, as we've seen, are super informative and remarkably helpful for understanding them. That common ACE ending is often your first big giveaway that you're dealing with an enzyme. Names like phosphatase, carboxylase, and oxoreductase tell us a lot about their job. They hint at the substrate the enzyme interacts with, its specific molecular partner, or they describe the type of reaction the enzyme helps to catalyze. It's like a secret code, but now you know how to start cracking it effectively. Understanding an enzyme's name is your first major clue to its function. Think of it like reading the title of a fascinating science book or a movie poster. The title gives you a really good idea of the story or action you're about to discover inside. Once you understand the meaning packed into the enzyme's name, a whole world opens up. You can then confidently look at the reactants and the products involved in its reaction. You can start to figure out exactly what chemical change is taking place under its influence. Are chemical bonds being broken, or are new ones being formed between atoms? Is the molecule changing its overall shape or its chemical composition? The name guides your investigation. So by taking the time to understand how enzymes are named, you unlock a wealth of information. You gain the power to predict what a particular enzyme is likely to do in a biological system. You can better understand its specific role in a larger, complex chemical pathway or metabolic network. It's a truly fundamental step for anyone studying biology, medicine, or chemistry. This knowledge helps us see how these tiny, incredible molecular machines work tirelessly and precisely to keep all of life going, from the smallest microbe to you. Enzyme naming is the key to unveiling their many secrets and their vital roles. Consider the following. Science is all around us in every breath and every heartbeat.